Hello.
Hi.
Hi, how are you? So, yeah, hello. Hello, Harshit. How are you?
Hello, sir.
Hi. I know you wanted to give a presentation on your project, so we'll do that. But before we do that, are there any updates? All right.
Hello, sir.
Hello, how are you?
Hi, ma'am. Sir, I am currently BTech student in second year.
Okay.
I'm from India. I am studying in Vellore Institute of Technology, and I want to participate in GSOC. And I want to participate in machine learning.
All
So
right.
I wanted to meet you.
Yeah. Well, welcome. Yeah. So that's great. Yeah, we'll be making decisions in the next this week, actually coming up. So this week we'll
Yes.
be sending out the acceptances. I have no idea how many slots we'll get from GSOC. And
Yes.
if you don't get selected, you can still work on a project. Just let me know and we can set something up because we always have people working on projects in the group, and they work at their own pace if they're not on GSOC. If they are on GSOC, then they work according to their schedule, and sometimes people continue on afterwards, and so there are a lot of
Yes.
opportunities. Yeah, so did you want to present?
Yes, I would like to
All
present.
right, go ahead and share your screen. Yes, I can see your presentation.
Sir, my project was about graph neural network in a GSOC problem statement.
Okay.
It it was a large project according to that given in the problem statement. So I have worked on GNN earlier. Like I can show you one of my projects.
Okay.
I read this research paper for making that project. I am currently working on a research project which is related to fluid mechanics.
Okay.
I can show you. That I will show you. So, sir, it is related to like edge focused and node focused network.
Okay.
In edge focus, the data is passed through edge, and node focused data is passed through nodes directly. Edge focused mainly focuses on specific types of which are specifically should be shared through edges only, like we can see in this research paper. Sir, I will show you one second.
All right.
So what happens is we can do recursion, like in fluid mechanics project. What we have done is we have a one node. Nodes are nothing but neurons there, and those neurons can transfer their data from one to another, and the recursion happen. One edge shares its data to nearby edges, and that shares to early others, and that come to a specific node one by one after recursion, sir. Sir, this is a research paper that I have worked on. It is related to fluid mechanics. It is about a probe design which is increasing its volume with time. We want to increase its volume in such a way that its buoyant force remains stable in water or any fluids available. So we are trying to predict how much volume it should expand according to the live conditions after the chemical reaction happening inside.
All right.
And one more project that I have worked on is about urban heat island monitoring systems. Urban heat island is, as the name suggests, heat island. Island means some area covered by water, and heat island means a area has more heat in itself than its surrounding. So that surrounding area is cooler and that specific area is hotter. That is called urban heat island. And for predicting those areas, we are telling which features like elevation of that area and other features like air quality and other. We are telling which feature contribute more to that area specifically, and we are doing same for larger area as well. I have learned Google Earth Engine for doing so, sir.
All right.
I have a video as well for that.
All right. Oh, yeah. Oh, you run that team, yeah. So are you using a graph neural network on this, or is just a separate project?
Sir, graph neural network project is a separate one.
Oh, okay.
For that, I have made easier that one as well. Graph neural network is more complicated.
Yeah.
We can make it easier. Yes, sir.
This is the Google Earth engine, though. So this is like a, yeah.
Yes, sir. It is for Earth satellite data we can use on Google Earth Engine. We have to learn it for using its data, sir.
Yeah, that's great.
Yes.
Yeah.
Sir, is video audible as well?
No, but that's okay. It's hard to. It reverberates too much. Yeah.
Sir, I can make website as well. I know React.js, Express.js, Django as well.
Yeah.
And sir, like I have a project also on React.js. I have a website deployed as well.
Yeah.
On GitHub. So, sir, these are my projects that I've worked on.
Yeah. Did you have a paper that you were going to bring up as well, or you had the? I see a tab. It says graph neural network. That one, yeah.
Yes, sir. This is yes.
So
this is a paper that I read to make that graph neural network our work, sir. And our paper is also there. That I can't show, sir, because it's in publication process.
Yeah.
So, sir, this paper tells us very well about how graph neural networks work in two ways: node focused and edge focused. Like this shows how fluid mechanics node focused and edge focused, and the data is transferred from nodes to nodes. Specific type of data is transferred through nodes and specific through edges.
Great.
Like for example, in fluid mechanics, we can see that, sir, like advection net is for calculating acceleration, and then collision network, then pressure network. Two of them are used in node focused and one is edge focused.
Yeah. What's the advantage of using? What's the advantage using a node focused versus an edge focused, or vice versa, I should say? Because we kind of know what the advantages of a node focused network are, but how would an edge focused network help deal with a problem of flow? Is it the interaction that you're capturing, or is it something else?
Sir, it is used pairwise. Edge focused is more specific for pairwise inputs. Like one input is dependent on another neuron. In case of node focused, it is not dependent on another. It is going directly. And in case like when collision happen in fluid mechanics, then that is dependent. Then we use edge focused. And in other case we used node focus for pressure and all other stuff.
Yeah.
So that is how we use it, and this makes it faster. This makes GNN faster.
Yeah.
If we separate both, if we include all together, then it will be much slower than normal. This one.
Yeah.
It is for accelerated Lagrangian fluid simulation.
Yeah. I see.
Yes.
That's good. This is the graph here, graphical abstract. Could you click in on the graphical abstract there?
Yes, sir.
Yeah, that one. Yeah, the graphical abstract is, right there.
Yes, sir. It is advection at first, which calculates acceleration, and then it is acceleration data is transferred to collision net, then collision net data is transferred to pressure net. And when we separate them, it becomes faster. In case we calculate all together, then it is much slower. If we separate them and gets a predicted output, then we use that predicted output and feed into another neural network, then to calculate another part of neural output, then that feeding to another, it becomes faster.
Yeah. Yeah, it looks.
In case yes.
So you do this for every time step. You evolve it.
Yes, sir.
Okay.
For every single node, we have to do.
Yeah. All
The
right.
more number of neurons, the more time it takes.
Right. Right. Well, that sounds good. Yeah. So this is actually an alternative to numerical methods. And
Yes.
yeah, we've talked about that in some of the work we're doing. We're interested in biophysics. We're interested in these tensegrity structures where you have the edges are important, quite important, maybe more important than the nodes. And so it's interesting to see some work on that.
What was the paper reference again? I just missed it.
Yes, I can share it.
Yeah. Here we go. Good.
Oh, thank you.
Yeah, that's great. Thank you, Harshit. That's it's an interesting approach, and so.
Yes.
Any other questions about the work there, the approach? Okay. Yeah, thanks. So let me share my screen now.
Yes.
Yeah. All right. So, yeah, we got a lot of things I want to talk about today. I have several articles, and then I like little pieces of news, I guess, and then a larger paper to talk about. So this is the first one I wanted to talk about here is this APS article on, it's an opinion article, That's Not Physics. And this is Andrew Zangwill, and this is, talking about things like emergent systems and complex systems. And this, is kind of a interesting topic in terms of complexity theory and how we apply it to things like biological systems. But it also kind of takes the perspective from the physics side, which is this actually physics or not? So it's a sort of a disciplinary boundary issue. But anyways, he asks the question, where do the boundaries of physics begin and end? The debate has persisted for more than a century. So this is the picture of a starling flock, which of course consists of a bunch of birds. They participate in this flocking behavior, which people have been able to capture using algorithms like Boyd's. And you can model this in a computer. You can also observe it in the sky, and we've been able to do a pretty good job of getting the details of these emergent structures down, at least in terms of the movements. We don't know a lot about like the actual behaviors in the context of these movements, but that's something that is less understood in terms of algorithmics. So this is we can replicate these kind of swarms and flocks and things like that. So this is a starling flock in a so-called murmuration or collective behavior of interest. And this is one of the many subfields of biological physics and biological physics itself that don't always belong in physics. So a lot of times, biophysics is, they're answering questions in physics, but they're also answering questions in biology, they're answering questions in computer science and so forth. So if you've been in physics long enough, you've probably left a colloquium or seminar and thought to yourself, the talk was interesting, but it wasn't physics. So this is usually a problem. I've had this problem in my career a lot where I've been in different departments and, you go to talks and they are very different from the rest of the talks in the department, and kind of situating work in different departments versus, in sort of an interdisciplinary group. And so a lot of times there's some, boundary issues with that. People have conceptual problems sometimes with thinking about things outside of their domain of expertise. So, if you get a biologist and a physicist in a room, sometimes the biologist and physicist won't really see this problem in the same way. So you'll have a flock, you'll see a flock in the sky, and the biologist will want to talk about certain things, and the physicist will want to talk about certain formalisms, and they're not quite on the same page, and it's because their training is quite different. And so one way that we've tried to overcome that is by doing this sort of cross-training, this sort of interdisciplinary training. But still you have problems of people recognizing it as part of a field or being a contribution to science. So this is, a problem in terms of promoting and hiring individuals. And this can be, just kind of it can shape the opportunities for people to work on different problems. So, in our group, that's not so much of a problem, but if you're in a more formal academic setting, it can be a very big problem. So, a lot of times questions that we've asked have happened in the context of, or sometimes in spite of, where their field of origin. So these debates over physics and what is physics and not physics are important. We've been doing this for over a century. A key moment for not physics occurred in 1899 at the second ever meeting of the American Physical Society. In his keynote address, the APS president Henry Rowland exhorted his colleagues to cultivate the idea of the dignity of physics, which basically means ask physics-like questions always. And that's of course as the next century would roll on, the nature of physics would change quite a bit, but that's the sort of attitude sometimes you get in physics or in any field, really. Much of the intellect of the country is still wasted in the pursuit of so-called practical science, which ministers to our physical needs, he scolded, and not to investigations of pure ethereal physics, which our society is formed to cultivate. So there is this aspect of like the pure versus applied research, and that's a different set of things where, you know, if you're applying your physics knowledge to a problem like swarming or flocking, you know, it's not viewed as real physics because it's not answering fundamental questions, even though it kind of is answering fundamental questions. But, you know, so this you know, has been an attitude that's persisted. Another, you know, a good example of this is acoustics. So acoustics started as a branch of physics, and it's become its own field since. You know, people have investigated acoustics in terms of its applications to, you know, sound systems. You know, there have been applications to cognitive science, applications to animal behavior, and so forth. So acoustics, which there's an Acoustical Society of America which studies, you know, the propagation of sound waves, the interpretation of sound waves by the nervous system, and so forth. That started as a physics endeavor, but now is this multidisciplinary field that takes in a lot of different disciplines and those perspectives. So it's interesting that acousticians, or people who studied acoustics, you know, in sort of the early days of that area, were given a cold shoulder at early APS meetings. At the time, which is the early 20th century, acoustics research was not uncommon in American physics departments. Harvard, for example, employed 5 professors who studied acoustics between 1919 and 1950. You got more people with World War II and the needs of things in that era. And then after the war, of course, you had other applied areas that started to pop up. So this is, you know, they were kind of like separated out from the field, or kind of expelled from the field, but they ended up forming their own sort of interdisciplinary field. It's kind of an interesting exercise in looking at how different academic fields kind of form and evolve. So, you know, cosmology is another example where it goes from being kind of not physics to physics. So in this case, cosmology started as not physics. It was very much based in astronomy, but then kind of came into physics with Albert Einstein's general relativity theory, kind of brought that into the field. And then there were other physics questions that resulted from that, which made it more integral in terms of the physics. And so not all disciplines have had such a clean path in and out of physics. So that's where we get to biophysics, where physicists apply their methods to the study of living systems. And so biophysics, which is probably the closest to our interests, you know, is kind of like skirted that line between physics and not physics. And so that means that, you know, we have questions that are maybe very physical in nature and questions that are very not physical in nature. And we have our techniques, which some come from physics, some might come from, say, computer science or some other area. And it's very, very eclectic in that way. And so, you know, it's an interesting kind of study of these different areas, especially with respect to biophysics. It's something in the chat. Okay, yeah, thank you, Harshit, for attending. If you need to leave, that's fine. But yeah, thanks for presenting on your project.
Thank you, sir.
Yeah. Okay. So this is another article. It's interesting. This is from Astrobiology. This is another topic that we're interested in. And speaking of things that are physics and not physics, this is sort of not physics, but it's also physics, if that makes any sense. So this is, again, astrobiology, which is, you know, sort of thinking about life on other planets or in other habitable zones other than Earth, and how that relates to life on Earth. And so there are a lot of interesting questions that have emerged from astrobiology. Astrobiology being sort of involving physics, but is also not physics. So this particular article is on the parallel evolution of gravity sensing. So this is where you have organisms on Earth, Ctenophora, Cnidaria, Placozoa. These are all invertebrates that have different structures to their phenotype. and but more importantly, they've evolved different mechanisms of sensing gravity. So an organism has to sense gravity. If you're in the water column, you have to know which side is up and which side is down. So if you're an organism that's differentiated into this asymmetric form, you have different ways of propulsion. You have different, sources where, places where food is, different food sources. You have to have some sense of gravity sensing so you can move to get to that food or move up and down the water column as need be. There might be chemical gradients that you need to conform to and things like that. So you need to have this gravity sensing ability. And in these three types of organisms, we've had three different forms of gravity sensing evolve. So if we look at this more closely, in Ctenophora, we have an arboreal organ with a statolith, which is this thing here. You can't really see it very easily. But it's basically this, there's this dome that has a statolith, which is a little stone. It's like an otolith in fishes. It's got these, accessory cells around it, and it has other structures around it. So it's this dome with these structures emanating from it. So it's one way to do this. That's one way that this was evolved in Ctenophora. Then in Cnidaria, which is a different set of organisms, we have the Cnidarian nematocyst. And so this is a little bit different, where you have the sensory epithelium here. You have this concretion, so again, it's like a stone, but it's not the same because it doesn't exist in a dome. It's in this layer of epithelial cells. You have this inner thin epithelium, sensory epithelium out here, and this structure out here, which I guess gets information from the outside world. And so you have this, again, like in fishes and in vertebrates, we have this thing called an otolith. And the otolith is in a structure, and this is like in humans, this is the inner ear. In fishes, they call it the otolith. So it's the structure in the head that allows us to sense, gravity and our sort of, when we move our heads or when we move our bodies, we can tell kind of the inertial sort of, we can register ourselves with respect to gravity. So this is the same thing here, it's just using a different structure. And then in placozoans, we have these crystal cells, which is, a type of cell where you have, it has a very different structure than these two. So these are called staticist architectures, and they do this in a different way in each set of organisms. So this is an interesting thing. Every organism has to deal with gravity, and there are different ways that you can evolve this organ. Obviously, it's important because you get these convergent evolution. This convergent evolution of the organ, how it gets kind of put in place in different ways in evolution. So gravity affects all living organisms. You need this for survival because you need to navigate your environment, but you also need to know kind of, even cells really need to have an account of gravity as they sort of form structures, but they don't really have as far as we know, they don't have like a specialized sensory organ for this. There's, you have chemical signals and physical signals that play a role in kind of maintaining the stability of the cells. And maybe, we've talked about like epithelial sheets where we have sort of a mechanobiology where you can build these hyper stable structures or structures that are resistant to the forces of gravity. So, but this is more about sensing gravity in the context of moving around the environment. So these are specialized organs for that. So, a lot of bacterial-sized microorganisms are subject to Brownian motion, and so are our cells in animal cells and plant cells. They operate under distinct microenvironment and viscosity constraints with a lack of detectable gravity sensation. So there are these different physical aspects of the environment, and you have to have this sensation of gravity to know kind of how to move against the gravity. And so many unicellular protists and multicellular organisms, which are less than 50 microns in size, have developed remarkable examples of graviperception, which is what they call sort of sensing gravity and responding to it. Gravity sensitivity often co-evolves with photoreceptors as one integrative system. So this is interesting that you see often in evolution, you see photoreceptors evolve along with gravity sensitivity. And it's worth thinking if you have like a organism, in like if we had organisms on another planet where they were, there was a very high gravity, what would those organisms evolve in terms of gravity sensation? Would it be a system like this? Or would it be much, much more complex? What would it look like? So this is, there's a variety of these kind of, organs and ways of sensing gravity. And we also have this sort of psychophysics that's a part of this as well. In many planktonic and benthic organisms, larval stages and land organisms, positive phototropism, which is the sensation of light, and negative gravotropism, which is the sensation of gravity. So a tropism is where you're moving towards a source. So if you have a phototropism, you're moving towards the photic stimulus. If you have a negative gravitropism, you're moving away from the source of gravity. So, this is where they can mutually influence each other. If you think about like different organisms in, say, the deep sea or in some other aquatic environment, oftentimes they're moving up and down the water column. And if they move up the water column, it's usually towards light, although it could be at the bottom if you're in the very deep sea, because you have, vents, volcanic vents and things like that. But you're often moving in the maybe against or in concert with gravitational forces. So gravitational forces pull down on the organism, and phototropisms might make you want to move up. So you need to evolve something that counters gravity, not only to keep yourself stable, but to move up the water column. So these are things that, all kind of co-evolve. Quantitatively, Hawk and Hayter, which they talk about up here, 2006, they state that light transduction appears to possess a logarithmic transducer, whereas gravitation or gravitransduction induces a linear one. So there are two different kind of responses to this. You have this logarithmic response, which is the response to light, and a linear one, which is the response to gravity. So that's interesting over a range of kind of values that you can get this response by the organism. So, in terms of gravity, if the gravity increases or if the light increases, there's a difference in the corresponding response by the organism. So, this is very important for both bioengineering and astrobiology. Understanding the diversity of these gravity signaling systems presents a unique reference paradigm to understand transitions of homology, which are these kind of independent evolution of different types of systems, to examples of convergent evolution within integrative systems. And so this is, I don't know where they put the citations for this, but this is an interesting article. And, there are a lot of questions in terms of astrobiology that, could be kind of answered by this paper. Okay, then the next paper here is this new paper on the path to complexity. This is in NPJ Complexity, which is a journal. It's again a Nature journal. I don't know what PJ means, but it's a Nature-based journal. So this is the path of complexity, and this talks a little bit more about complexity theory and kind of the origins of complexity theory, and how, kind of how this has grown around different problems and different questions that people have had, and how they've tried to answer them. So one of the things about complexity theory is it's a departure from the way that we answered physicsy questions or physics-like questions, or even not physics questions in the 20th century. So, in the 20th century, we used a lot of, statistical methods that weren't didn't capture things like emergence or dynamical systems. But in the later 20th century, these techniques have been developed that we associate with complexity theory. And there's a lot of, especially like kind of with the late 20th century, there was a lot of hype around it. And so Stephen Hawking was quoted as saying, I think the 21st century will be the century of complexity. So that may or may not be true, but there was a lot of thought that this would be the sort of the science, the unifying science of the 21st century. Well, so far I don't know if that's true, but I guess it's got its promise still, but also it's been applied quite a bit throughout science, so that's good. So complexity science studies systems where large numbers of components or subsystems, at times of a different nature, combine to produce surprising emergent phenomena apparent at multiple scales. It is these phenomena hidden behind the often deceptively simple rules that govern individual components that best define complex systems. So if we go back to our swarm, each of these birds are one of these they look like black dots, but they're a bird. They're an individual agent. They have behaviors, and the behaviors aren't always the same. And so, modeling this as like a set of particles, might be inappropriate. We might want to add in like individuality. We might want to add in like, different perceptual systems or different types of decision-making systems. We might add in some noise to counter sort of this idea that every agent executes the same set of rules. There are a lot of things we can add to the model like this. But we have to have a starting point. So we have this model that takes individuals, looks at their interactions, and then builds these large-scale structures out of it and says, okay, this large-scale structure is a different thing, and it, is different from the state of the individuals. Yeah, tensegrities could be thought of as a type of complex system. You could do that, yeah. Definitely, like a tensegrity structure is different than the component parts. So the component parts all contribute to this structure, which is a different state than the individuals. And so, there have been a lot of, this requires a sort of non-physics or not physics sort of interdisciplinary collaboration, studying a problem across scales and disciplinary domains. And so this is something that is, goes back centuries, but has really kind of crystallized in the latter part of the 20th century. So in 1962, Herbert Simon laid a roadmap for the study of complex systems in the architecture of complexity. Herbert Simon was from the social sciences, so he was doing a lot of stuff, post-war stuff with early artificial intelligence and organizational science. So it's very different from like the physics of a swarm or a flock, and the sort of biophysics questions that we ask. But the methods are very, you could have similar methods. They're relevant to both areas. A decade later, the physicist Philip W. Anderson addressed how this philosophy clashes with the standard reductionist hypothesis in a 1972 paper called More is Different, Broken Symmetry in the Nature of the Hierarchical Structure of Science. So this is a classic paper sort of that lays out this sort of initial approach to complexity theory as a field. And so the people from the Santa Fe Institute love to cite this as the foundational paper of complexity science. But there was a lot of things going on at that time that were kind of going in that direction. So now in this essay, Anderson argues for the need for multiple perspectives, since the ability to reduce everything to simple fundamental laws does not imply the ability to start from those laws and reconstruct the universe. So, you can have laws that sort of govern, say, something like emergence or emergent complexity. They're different than the laws that sort of govern the origins of, say, like life or the origins of a physical system or something like that. So, a couple of agents kind of mucking around operate by a different set of rules than a complex emergent structure. It's basically what he was saying. And you can't get there from looking at things using a standard reductionist approach. You can't break down the swarm or the flock or the emergent structure into parts. And so if we're modeling something, that's an interesting thing to recall, because if we model something, we're modeling a complex emergent structure a lot of times, especially in biophysics, especially in development. And so, taking these parts of the system, these cells or parts of cells, and modeling the whole is going to give you sort of a different set of principles that you have to model. So we talked about multi-scale models. And one of the reasons we want to build multi-scale model is because we have things like, macromolecules or cells that contribute to larger structures that are very different in the way that they operate. So we saw that last week with the platforms, the modeling platforms that we were talking about. They treat these different scales very differently in terms of their governing laws. So, there are different types of ideas that were floating around in the 1970s and into the 1990s. Along with this, there were a lot of popular books in complexity theory that came out around that time. It was a very heady time for that area. But, was it physics or not physics? Well, they had a lot of it in physics departments, but a lot of it was also non-physics, because you had, you were dealing with biology and you were dealing with social systems. And so it was very different from what people were doing necessarily in physics departments. So, so at the same time, John von Neumann and Stanislaw Ulam were developing theoretical machines, which became foundational to the future field of computer science, that could build functional copies of themselves with hopes that they would eventually evolve ever-increasing levels of biological complexity. So this is really like the sort of the beginnings. At the same time, in fact, probably from about the early 1960s on, you had people trying to build upon the self-replicating aspect of biological complexity and the generative aspect of biological complexity. So, we don't talk about genetic algorithms too much in the group. We talk about it off and on. But, developing those kind of models, the sort of replicator models, John von Neumann did some interesting things with cellular automata that, really kind of mimicked how biology and biological complexity work. So there were a lot of different kinds of arguments for self-organization, and genetic, working with genetic regulatory networks and other types of physical models for understanding self-organized criticality and things like that. So there was this explosion of work on biocomplexity that really kind of persists to this day, and they're very applicable. They're very different from what came before. So this paper is interesting because it kind of goes over a lot of the aspects of the early models that kind of came out, kind of emerged, pardon the pun, in the late 20th century. And then they make a point about it being interdisciplinary, not any disciplinary. So it doesn't require you to kind of just not have a discipline. There is very much an interdisciplinary aspect where you're combining things from multiple disciplines. And this is, again, this sort of, you might find this a trivial argument, but it's very important in terms of sort of focusing the work on things that people are expecting to see. So there's this focus on sort of interdisciplinary work, which means you're taking things from two fields. It could be like, physics or some non-physics field, and putting them together. And so this kind of talks a little bit about this new journal, or the scope of this journal. So I'm not going to get into that, but basically, they're kind of talking about this aspect of disciplinarity and how that relates to complexity science. Okay, so Susan said everything is physics. Well, I don't know if I'd go that far, but that was kind of the attitude, I think, of a lot of the complexity theorists of the late 20th century. So a lot of them came from physics, and so they said, okay, everything is a physics problem, but it's you also have to have these other methods through it. You want to study biology in a way that biologists haven't studied it, nor physicists have studied it before. But the techniques are from physics. So yeah, at the root, I guess the implication was everything is physics. But
yeah, it's sort of the baseline, and you can build actually from physics to chemistry, and from chemistry to biology. It's sort of and you can go directly from physics to biology. Yeah. At least that's the way I see it.
No, yeah, I mean, there's definitely like a line from physics out to biology, and because I mean we've had physicists in biology or physical biology for a long time, probably 100 years or more, but yeah, a lot of the techniques have developed in physics, both the quantitative techniques, but also the techniques like microscopy techniques, or DNA sequencing comes kind of from chemistry, but also physics, and it comes into biology.
Yeah, you can essentially describe a lot of biology using physics.
Yeah.
Biology tends to be observational and is certainly written differently than engineering or physics, because they'll say, oh, it's this way, I think, or I might be wrong. And you're, as an engineer, you're going, it might fall down. Yeah. So it's hard to read sometimes, because did they mean what they just wrote this whole paper about, or why did they say that?
Yeah.
It has been difficult.
Yeah. Well, yeah, Susan, you have this problem in your own work, where, well, in this case, it's engineering and not engineering, right? Like.
Yeah, I've run into difficulty. Oops.
Yeah.
I don't think I'm just going to turn this off. It's not lined up. Anyway, yes, and so I get asked by an engineer, is your construct right? Like for instance, the tensegrity that I'm doing.
Yeah.
And I say, maybe. And maybe I'm wrong, because that's the way it's phrased by the biologist. So then he thinks I don't know what I'm doing.
Yeah.
But yeah, this is my best guess from reading the biology.
Right.
And it's all observational, and I might be wrong, written right into their papers. So what am I supposed to say?
Yeah. Yeah. Yeah, it's definitely, it just persists, I guess. Yeah, so I had posted something in the Slack. This was a paper from iScience. It's interesting. We talked about reservoir computing a couple weeks ago. This is Jack Jasinski and some co-authors. This is on modeling non-genetic information dynamics in cells using reservoir computing. So they're doing some interesting work with non-genetic information in cells. And this is, I pulled this paper up, put it in the Slack. And this is the paper here. So they first of all, they introduce a novel theory of non-genetic intracellular information dynamics, which I assume is like things moving around the cell. You have a lot of things like going on in the cell, but I think in this case they're focused on energy-driven ion-specific membrane pumps. So they're trying to maintain these transmembrane gradients, and so they have these information dynamics. And then they say information is transmitted by ion fluxes. across membranes and along the cytoskeleton. So they take a certain part of the intracellular milieu and they sort of focus on that. They're focused on the information transmission there. Then they take that and they use a reservoir computing model based on the cytoskeletal network. So again, this is a network approach, but it's a reservoir computing approach. So the reservoir computer that they build is taking like the cytoskeletal network and sort of throwing basically turning it into a random network, and then they try to model it as a sort of trying to find like the patterns or modes of information transmission or spatiotemporal information distribution. So this reservoir model demonstrates efficient spatiotemporal information distribution. So this is their graphical abstract. And again, this is, these are very useful. I like how there's been a trend towards this. So this is the non-genetic communication mechanism necessary for rapid cell response. Basically, if you have a cell that has electrical activity or, electrophysiological activity, there's this influx of ions, and there's this, the signaling that happens. So, briefly, you start with a resting cell. You have this extracellular perturbation, so you perturb the cell in some way. There's some transmembrane ion flux, so ions can come in, but they can also leave. And there are different types of ions, so they're different, there's potassium ions, there are sodium ions, and so forth, and you can go through. And so you can model the influx and outflux of those. But this is also, it's not just happening in isolation. There's this information flow along the cytoskeleton that allows for this transmembrane flux to occur. So the cytoskeleton essentially has to remodel to allow for this. And then there's this overall cellular response. So as there's information flow along the cytoskeleton, there's a whole cellular response that results from that. So that's what they're getting at, this kind of model. And they're using reservoir computing to sort of get a handle on this. So this picture down here is modeling intracellular information dynamics and cellular decision making with reservoir computing and deep learning. So you have this model of the cell, this 3D model of the cytoskeleton across the cell membrane. You model this as a reservoir computer, and you come up with these components. You have this model of information processing where there is this inhomogeneity along the cytoskeleton. And so you have like all these different parts that you have to model in this network: the cell membrane, peripheral cytoplasm, the cytoskeleton, the nucleus, plus endocellular reticulum, plus mitochondria. Those are all like one class. And then information signal. So the information signal is in white, peripheral cytoplasm is in red, cell membrane is in blue, the nucleus, mitochondria is all one thing, and that's yellow. And so they use those as sort of the nodes of this reservoir computer. They throw them and they have these randomized connections, and then the reservoir computing, as we talked about in previous meetings, kind of makes sense of this. So that's what they're doing in this paper. I think it's an interesting approach to this problem. Again, reservoir computing, you could argue that it's kind of this sort of magic that happens. Because, we know that, like, we can model, we know kind of how neural networks work. We know how deep nets work. Sometimes they're hard to interpret what's going on inside of them. And the same if not more, holds true for reservoir computing. So this is where they talk about Hodgkin and Huxley and some of the models that we have currently. So in Hodgkin and Huxley, we model the ion channels all as sort of from different classes with different classes of ions, and we can model the flux depending on what's present in the cell. And you can model things like you model cellular depolarization that way, but you can also model action potentials that way. So there are a lot of ways you can apply Hodgkin and Huxley. We hypothesize that ion flows in neurons represent a specialized adaptation of a broader information acquisition, processing, and response dynamics centered around the cell membrane. So Hodgkin and Huxley doesn't model the information content, it doesn't take into account a lot of the structures in the cell. It just models a flux of ions. That's all it does. But in this case, what they're modeling is they're modeling the cellular response, the phenotype. And so this is, I think a nice addition to Hodgkin-Huxley, and it tells us a lot more in some ways of what's going on in the cell during these ion fluxes. So that's that paper in iScience. All right, any questions or comments? Okay, so again, for the benefit of repetition, Google Summer of Code, we're going to be making decisions this week, or INCF will be making decisions this week, and we're going to live with the decisions they make. So, if you get selected, you'll get a message sometime, I think, midweek. And if you don't get selected, you can still participate in the summer. Once we get our people coming on board, we'll have an onboarding process. We'll have what they call the community period, which is several weeks of getting to know the community. And I'll try to make that easy for people. Now, in our case, we have two communities, or two sort of allied communities. We have DevoWorm, which is this group, and we have OpenWorm, which is the larger group of OpenWorm, where we have people doing other kinds of activities, modeling other parts of C. elegans. And we talked about this in previous meetings. And so people are welcome to do things with graph neural networks, with DevoLearn, with anything that we're doing in the DevoWorm group, and then anything OpenWorm is doing. So we have a lot of things that you could do, whether you get selected or not. And if you're doing your project, you want to stick to the schedule on the project, but you also want to maybe make some connections in the larger OpenWorm community. That would be good, I think, for your future endeavors, but also for maybe some of the work you're doing in the projects, because our projects, oftentimes in past years, we've had people want to model things like movement of the worm or other aspects of worm behavior, biophysics of the worm. And there are tools in OpenWorm's pantheon of tools to do that, and their data sets as well, so we can investigate those in the community period. But even if you don't use them, it's nice to know about them. So that's coming up this week. So yeah, next week we'll continue with more things. Probably welcome our new students for GSOC when we find out who those are. So thanks for attending, and talk to you next week. Thank you. All right, now I'd like to talk a little bit about a technique called TOMO-Seq. So this is a technique that looks at spatial transcriptomics. And, there are a lot of advantages of spatial transcriptomics, especially looking at things like embryos and even individual cells, where you can look at the transcripts happening in a certain region, and you can localize those transcripts and see their changes over time. So it's very useful, but it's hard to kind of make sense of the data. People use things like dimensionality reduction, UMAP, and so forth. And there have been some debates as to whether UMAP is useful at all. Now I happen to think it is, but I think often people misinterpret what exactly is going on. So we're going to talk about some techniques for this, a book chapter on the technique. We're going to leave the analysis and the methodology for another day, but I just want to go through this method now. So this is from a book, Methods in Molecular Biology, number 1920. Vertebrate embryogenesis, embryological, cellular, and genetic methods. So this is a book has a host of methods in it. The method itself is in chapter 9. This is RNA tomography for spatially resolved transcriptomics, or TOMO-Seq. So they're using RNA tomography, and I think we talked about this in a paper last week and I wanted to follow up on it. But it may have been a talk I attended too where we talked about this. But anyways, this is using imaging to find RNA in a cell. So this is really, kind of an interesting technique, especially with respect to, other structures. So if we're looking at cytoskeleton, or we're looking at the shape of a cell, or the shape of a population of cells, we can use this in tandem to find RNA expression and resolve that spatially. So the abstract reads, embryonic development is heavily dependent on temporally and spatially restricted gene expression. Spatially resolved measurements of gene expression are therefore crucial for identifying novel regulators and an understanding of their function. So we have in situ methods for this. We can use like different antibodies, and we can stain for RNA, and we can look under a microscope and see the sort of where those stains are in the cells if we have like cell culture, or if we have a model organism, we can look at like a slice under the microscope. We can see how it kind of, where it's expressed. And so this, this is good for like looking at specific genes if we're interested in a gene of interest and how it's expressed, maybe across different time points or with respect to some, dysregulation or aberrant expression or some sort of function. But in situ methods do not resolve global gene expression. And sequencing-based methods usually do not provide spatial information. So we have these in situ methods that are, of course, pretty established. We can't necessarily know much about global gene expression. We can only know about maybe a gene of interest or several genes of interest. And so, in a high throughput context, that's not very useful. On the other hand, we do have next-gen sequencing, and that does provide this global gene expression information, but it doesn't usually provide spatial information. So something like RNA-seq, we get an abundance of transcripts, we get a sequence for different segments, so the RNA has different sequences that can be mapped back to a reference genome, but we can't necessarily know its spatial location. If we do single-cell RNA-seq or single-cell sequencing, we can get down to the level of a cell, but beyond that, we can't say within a cell or even within a tissue where these RNAs are being expressed differentially. Because in fact, we can take single cells, but we often take a sample. We dissociate the cells from its tissue, and then we take the single cells themselves, and we usually average over single cells if we want to say something about a population. So we don't have that spatial context. Here we describe TOMO-Seq, a method that combines classical histological sectioning of embryos, which is the thing that we're doing in situ methods, with a highly sensitive RNA sequencing technique. So they're taking RNA sequencing, they're combining it with the histological preps of in situ gene expression assays, and they're using this as a single method. So in this paper, in this chapter, they're applying TOMO-Seq to zebrafish embryos, and it allows reconstructing the spatial gene expression of thousands of genes. And so, when we talk about spatially restricted gene expression, we're talking about the establishment of patterns that sort of play a key role in the development of tissues and organs. So when we're talking about spatially restricted gene expression, we're talking about pattern formation, we're talking about differentiation of cells into different tissues, we're talking about spatial phenomena, we're also talking about temporal phenomena. But we can view this in terms of space. So, whatever processes happen in time, they become manifest in spatial arrangements. And so, the gold standard to visualize RNA localization has been whole mount or section in situ hybridization. And these are simple protocols, but they don't give us the kind of information we need for what we want to do here. So, there are a lot of techniques like mirrorfish and seqfish that have been developed to overcome the limitations of in situ, which has been just basically the number of genes that you can look at any one time. by sequential hybridization of short probes coupled with fluorescent dyes. Some 100 genes and their localization can be assessed in a single tissue section. So these are these FISH methods. This is, again, it's getting better, but it doesn't tell you a lot. Like, it doesn't square away with a lot of the next gen sequencing. While providing detailed spatial information, these protocols put high requirements on the equipment and currently do not scale the level of whole transcriptomics. And then, of course, classical RNA sequencing provides transcriptome-wide gene expression data, but spatial information is lost. So here they describe TOMO-Seq, a method for spatially resolved transcriptomics, combining RNA barcoding techniques, which is used in single-cell approaches, with classical tissue sectioning, which is, of course, used in situ. And, of course, TOMO-Seq doesn't require any real specialized equipment. It just can be used. You can use standard lab equipment to do this. They provide a protocol in this chapter. So the steps from the fresh sample to the sequenceable library can be accomplished in 5 to 7 days. It's a pretty good timeline for doing this. Where a lot of techniques will take weeks and weeks to perfect. I mean, I don't know, depending on whose hands this is in, how long this will take, but ideally it will take 5 to 7 days. TOMO-Seq is a simple but powerful technique for accessing spatially restricted gene expression and is particularly suitable for applications in developmental biology. The spatial expression of thousands of genes can be mapped simultaneously and consequently can be used to describe animal development. So the sample preparation, it really doesn't involve very much stuff. You need polyacromide beads, you need the typical lab equipment, you need trizol, glycol blue. These are all things to extract RNA from tissues, from cells. So, it's not that much more than your typical sort of lab equipment. And so you just need to extract the RNA from a sample. You need to run cDNA synthesis and, basically some PCR, and you need to be handy at PCR to do this, to run this protocol. And so what they're doing is they're working with zebrafish embryos. They're using a cryostat to get thin slices, and they're using a slightly modified version of the CellSeq2 protocol to prepare sequencing libraries of slice-specific DNA barcodes. So that's citation 8 and citation 9. So this is CellSeq2. This is a specific protocol for this. And so basically you're taking a tissue sample, you're extracting the RNA, you're building a barcoding library of cDNAs. You're then doing all these steps that involve like amplifying RNA and checking for quality control, and then building a PCR library check and sequencing. So you're doing this, you're going through this protocol. They introduce these stop points just as a means to do a reality check to make sure that everything is working well. So you're actually creating these cDNA libraries that can be used then to match up with the RNA. And so then we're talking about spatial resolution. So we want to look at the zebrafish larva at 48 hours. So naturally, if we think of the zebrafish embryo, we have this spatial relationship between the anterior end and the posterior end. We have this, we're trying to sequence different tissue slices, so we're taking the slice from the anterior to the posterior end. We're kind of mapping out what this, where these slices belong along this anatomical axis. They're going in the direction of sequencing. So we're sequencing from anterior to posterior. So in terms of output material, we have these cDNA libraries. We're matching them up to RNA. And this is the output material. This is a spectrum for aRNA. So this is where you have typical aRNA on an RNA pico chip. And this is a typical library in a DNA HS chip. So this is in base pairs, and you have all these samples. And you want to get a sense of how many bases long are the different fragments. And so we have this distribution here. We want to be able to match this peak of between 3 and 500 base pairs, and we want to be able to minimize the number of over 1,000 base pair size fragments. So we want to find fragments that are small enough to sort of identify unique RNA, but we want things that are also clean. We don't, we want to have a good sample, good representative library. And so this is an example here of aRNA. This is sort of in between 3 and 500, minimizing everything over 1,000 base pairs. This is the final library size distribution on a bioanalyzer high sensitivity chip. The distribution should have a peak between 3 and 500 base pairs, like the first figure, and fragments larger than 1,000 base pairs should be negligible. So we have this peak at about 1,300 base pairs. So, we can actually also look at artifacts. So this has a smaller peak around 150 base pairs. And so this can be due to primer artifacts, which is basically, in the step where you're amplifying your RNA, and you can filter those out. That's not a problem. But you want to have a clean curve. And sometimes you have messy curves. Sometimes you have artifacts. So those you want to be able to check for that. And so this is what the data should look like here. So we have our genes of interest here. We have, again, this is not, a candidate gene approach. This is where we're getting these fragments and they can be mapped to a specific gene. And so then we have this basically this list of genes, although it's not a candidate gene approach. We just have our different fragments representing what that gene, the constituent of that gene. And then we have our sections. So we have, for example, the gene ANK1, and then we have section 1, section 2, and so forth. And so recall that we had that figure of the zebrafish embryo where we go anterior to posterior. So section 1 would be anterior, section 96 in this case would be posterior. And then there's a number, which is sort of your measure, your target measure. And so what you're measuring is the presence of ANK1 in each anatomical section. So you're taking each section, you're getting not only for ANK1, but for all your genes in your data set. And sometimes, you have fragments that don't map to a certain gene. So that's what these dots are. And sometimes you don't get amplification, you don't get any sort of signal. So you have these dots here. There's missing data in here. The dots can be also, this is sort of showing like where, you have, in this case, you have, so that's, and then there, we have to clean up our data and things like that. But this is what the output should look like. You should have different sections, you should have different genes, and then the intersection of those two. And so, we're using a measure of normalized expression here. So our target measure is where we take the raw number and we normalize it against some housekeeping gene or some other method of normalized expression. There are a number of methods of normalizing gene expression. We typically don't want to take the raw number because the raw number doesn't really mean anything. We can't compare it to anything, especially across sections. So we usually normalize it against something. And so this is, on the right, this is a graph of normalized gene expression against section number. So here we have our section number. Again, section 1 is anterior most, and then the highest number is posterior most. And this is for one gene, EXD2, or at least transcript that belongs in that gene. So this is spatial expression of XD2. This is the normalized expression, and you can see that in basically what this graph is telling us is that this gene is expressed largely in the anterior portion of the embryo. And so you have some expression down in the posterior end. That could be noise. That could just be, random gene expression, whatever. But we can say basically that there's maybe something going on in the anterior end that's interesting that involves XD2. And then since we have all of our genes and we're assaying them in the same context, we can then put together maybe like a gene regulatory network. and say, okay, what is XD2, what is it related to? What is it in a network with? What is its function? We can use all sorts of bioinformatics tools to augment this method and ask questions like that. So we can generate a number of graphs for different genes and different RNA sequences, where we can say, all these are upregulated in the anterior portion of the embryo, so there must be something going on there. So this is how we can go from this kind of, I wouldn't say it's high throughput data, but it's kind of a shortcut to a lot of the uninterpretable big data of next-gen sequencing. So this is an interesting approach. It certainly is, I think, a complement to maybe next-gen sequencing approaches. It certainly also gives us really useful information that we otherwise wouldn't have. And so that's all I wanted to say about that paper. We'll probably talk more about this in future meetings, so thank you for paying attention, and I hope you learned something.